topological structures and certain network properties. Unstable network with different topological structure and network properties. So network pharmacology is proposed based on the theory of network biology and biological balance, and thus it provides a new idea for drug discovery as well as understanding the mechanism of drug functioning. So different types of network, as I said, someone are working on gene regulation network, some are working on protein-protein network, some are working on signal transduction network, some are working on metabolic network. So it depends. Now, very important slides. What are the different common databases and tool which we are using in the network pharmacology? So in network pharmacology, first step is, if you want to do, first you have to identify the targets of the compounds. So to identify the target of the compounds, we have a different databases. So what are these databases? We have Swiss target predictor, C database, Stitch database, drug binding database. So up to this drug binding database, these are the databases which are freely available. Every one of you can access, just do the Google. And these are the databases which help us to identify the target of the compounds. Compound can be anything. Compound can be your new chemical entities. Compound can be your existing drugs if you are repurposing. Compound can be your herbal molecules, whatever it is. So that is the first step. And second is, you have to be identify the target which are involved in that particular disease. Suppose you are planning to work in cancer, diabetes, and so on. So you have to identify the targets which are involved in disease. And to get this, these are the different databases: DigiNet, GeneCard, OMIM, HPO, and Malacard. Now, next step is you have to be identify the common target between the compounds and the disease. So Vani tool is the one of the commonly used tool which will help you to identify the common targets among the compounds and the disease. Now, next step is you have to check the interaction among targets. So to check the interaction among targets, these are the different databases: String, BioGrid, Deep, Stitch, Inect, and all are freely available. And Cytoscape, maybe many of you have heard about Cytoscape. Cytoscape is the one of the software which help in the visualization of the network. And SR plot is the pathway involved or enrichment. So now what are the steps? If you want to do the network pharmacology, what steps you have to be follow? So first step is, as I said, you have to identify the targets of the compounds, new chemical entities or phytoconstituents or herbal, whatever it is. So to get that, commonly used databases is Swiss target predictor, C database, Stitch database. If you go through that databases, you will find that they will ask you to provide a smiley notation. So smiley notation of the compounds, you can get it from the PubChem, you can get it from the Campbell as well. So Swiss target predictor, what most important thing is, before using the database, just read about the database first. There are a lot of information is available on the web page of the database. Just go through it, and then you can use it in a correct way. So Swiss target predictor estimate the most probable macromolecular target of small molecule available on this link. The prediction is founded at the combination of 2D and 3D similarity with the library of these many compounds. And 2D and 3D similarity. 2D similarity is quantified by the Tenmoto coefficients between the fingerprint vector, that is FP2, of both query and the screened molecules. And that 3D similarities you can calculate by ES5D of conformer. And these are the threshold values which are there in the database. The threshold value are set at 0.85 for ES5D similarities and 0.65 for FP2 similarity. So below such threshold, the known active are not similar enough to be displayed. So this is the cutoff you have to be keep in the Swiss target predictor. By default, you will get it, but you have to be checked as well. So probability of 1, if you are getting, then it means what? It means the most probably the query molecule is a part of your known active screened molecules. So this is a web page of Swiss target predictor. Just type in a Google Swiss target predictor. You will get this. You will click on this, it will open like this. And it's very easy to use it. How to use it? You just add the smiley notation of the compound, or you can draw the structure of compound here. And then click on the predict target. You will get the target which your compound can be hit. But of course, when you are using the Swiss target predictor, you have to be very careful. This Swiss target predictor contains the data of these 3 species only: Homo sapien, Mus musculus, and Rattus norvegicus. If you are planning to do experiment in the rabbit based on your network pharmacological results and you have used only the Swiss target predictor, you can't extrapolate it, because Swiss target predictor is providing the data of only these three species. So that's why, as I said, to read about the database is very important when you are planning for a network pharmacology. So it's very usually user friendly softwares or databases. You just click on predict target, you will get the result like this. You can export the results in a different format. Most commonly, we are exporting the results in a Excel form. So you have to click on the CSV and you will get such kind of file. In that file, you can see these are the targets which your compound can hit. These are the common name of the target. These are UniProt ID. These are the Campbell. These are the target class, and these are the probability. And these are the known active that I shown you 3D by 2D, right? Our next database is C database. So now maybe question arise in your mind, why we are using different databases for the same purpose. This C database is also used to predict the possible target of the compound. So why we are doing same thing in a different database? The reason is we are using freely available databases, and these freely available databases are not updated time to time. If you go through their websites, you will find that maybe they last updated in 2020, maybe it's updated in 2019, whatever it is. So to make a comprehensive, we have to make a comprehensive file so that we can't miss any targets. So you have to search all the databases, and after searching all the databases, also you have to do the data mining of literature. Maybe recently in Nature, some article has been published and they have identified new target which your molecule can hit, or new target which is involved in disease. So you have to include in your Excel file as well. So this is another database which you have to search. It's a freely available, but it's provided by the Department of Pharmaceutical Chemistry at the University of California. It's free of charge, but they have, if you go through the database, they have mentions you can use this database at your own risk. So it means that they are not taking any guarantee that your molecule will means anyone can know about your molecule. So they are mentioning you can use it at your own risk. So if you are working on a very confidential new chemical entities and you have not patented yet, don't use such kind of databases, right? These are the freely databases. When you are putting any structures, anything, so it will be enter into that one, right? So that is your C database. Okay. Just 1 second. Hello. मैं उसमें ऑनलाइन लेक्चर में ऑनलाइन लेक्चर में So this is the web page of the C database. You just type in a Google C database. You will find this. And again, it's very easy to use it. You have to just add the smiley notation of the compound. Click on the try C. You will find the results. Then another database to identify the possible targets which can be hit by your compound is the Stitch database. So this is the link for the Stitch database, or you can put, you can write only the in Google Stitch. So this is a database of known and predicted interaction between chemical and protein. It contains data of genomic context prediction. So if you are planning to work with genome interaction, gene interaction, so you can't miss such kind of databases. And this database is freely as well as commercially available. If you have a money, you can purchase the commercial version as well. Otherwise, you can use the free version. So it provides a combined score, that is combined means integrating the probability scores, probability from the various different types of evidences. And this is the web page of the Stitch. You just type in a Google Stitch, and just you can see it's again very easy to use. You can just add the smiley notation of the compound, or you can upload the structure of the compounds, and here you can select the organism. So for and selection of organism is depend upon after network pharmacology or computational approaches means in what type of experiments you have planned. So accordingly you have to be selected. If you are planning to do a experimental study in the rat, so you have to select rat norvegicus from here. If you are planning to do the experiment in the mice, so you have to use the mice species, right? So you have to click on the search and you will get the results. So another database that is to predict the possible target of compound is Binding DB. This is the School of Pharmacy and Pharmaceutical Sciences at the University of California. This is a public web accessible database of measured binding affinity, focusing chiefly on the interaction of protein. Consider to a candidate drug targets with the ligand that are small and drug-like molecules. So this is the Binding DB. And again, it's very easy to use. You can just type the name of the compound, like in this case, you can see that we have typed the rutin. Rutin is one of the phytoconstituents, and click on go. And after that, you will get the targets. So next step is identification of the disease targets. So I hope, but before these steps, after these steps, so you have to do the data mining of literature as well. Now, next step is identification of the disease targets. What are the targets which are involved in that particular disease? So, to identify the disease targets, the DigiNet is the most popular databases which you will find in every network pharmacological paper. And DigiNet Plus is the another version of DigiNet which is for the commercial purposes. But DigiNet is freely available, where you can download the targets which are involved in that particular disease. So this is the DigiNet, and DigiNet Plus is the this one, particular commercial one. This is the web page of DigiNet. Just type in a Google DigiNet, you will find this web page. Just go through it and you can create your sign up login ID and password. Just create it like that. We have created, this is my login ID and password in the DigiNet. And after that, you can click on the search, type the name of the disease, and click on the summary of gene disease associations. You will get the file in which you will get the targets which are involved in that particular disease. Like if you are planning to work in a diabetes, so you can add the diabetes. If you are planning type 2 diabetes, or if you are planning to cancer, so you like that, you can download the targets which are involved in that particular disease like that. So you can see in this case, we have downloaded the targets which are involved in the drug resistance epilepsy. So you can get it. These are the targets. These are the genes which are involved in the drug resistance epilepsy. So like that, there are other databases to identify the targets which are involved in disease. That is a gene card. So gene cards, you can see it's developed in the Israel, and comprehensive and easy to use human genome annotation database, and it's provides the relevance score. So in gene, this is the gene card web page where you can add the name of the disease. Like we have identified the gram-negative bacteria. So in gram-negative bacteria infections, what are the targets? So you can see these are the targets which are involved in the bacterial infections in human. There is another database, man OMIM. OMIM is another comprehensive database, so, it's updated daily. And this was created in 1985 by collaboration with National Library of Medicines at John Hopkins. And from this database also, you can identify the possible targets which are involved in that particular disease. So this is the OMIM database web page where you can use it. It's also freely available. Recently, it's completed 50 years as well. So this is very important database if you are planning to work on genetic disorders, so you don't miss that database. So now next database is your HPO. Just 1 second. Hello. Good afternoon. Yes, yes. Yes, yes. Yes. Yes, sir. Yes. Okay, no issue. Yeah, no, sure. Thank you, sir. Sorry. So there is another HPO. HPO is a human phenotypic ontology database, and it's established in 2007 in Cherry University Hospital in Berlin, and provide information on medical related phenotype. And NIH supported the project that integrate, align, and redistribute cross-species gene, genotype, variant, disease, and phenotypic data. So HPO is also a very important human phenotypic ontology. So this is the web page of HPO where you can type the name of the disease, you will get the targets. It's very easy to use, Malacard. Malacard is another human disease database available at this, integrate both specialized and general disease list. And also beauty of Malacard is it contain the informations or targets of rare disease. So I hope everyone of you knows what are the rare disease, genetic disease, complex disorder, and more. So you can't miss the Malacard. So this is the web page of Malacard. Just type in a Google Malacard, you will find this. This is the human disease database. Then, as I said, now what step you have done so far? One step you have done, identifications of the compound target, right? And second step, which you have to do the target which are involved in that particular disease. And you have used various databases to do that. So what you have to do, you will get different Excel file from different databases. So after that, you have to make one comprehensive Excel file. And it's quite obvious that when you are searching the same thing in a different databases, there can be duplicate as well. So you have to identify the duplicate in the advanced Excels and delete it. and make one comprehensive file, right? So now you have a two Excel file. One Excel file will contain the targets which your molecule can hit, and another Excel file will contains the target which are involved in that particular disease. So disease will be depend upon your research area, right? So that is the two files which you have be prepared comprehensively. And as I said, the major limitations of freely available databases is they are not updated on regular basis. So that's why we have to do the data mining of the literature as well. So you have to do the data mining approaches. You can Embase is commercial one, but Medline is your PubMed where you can do it. Clinical trial websites you can search, and there is a proper search items you have to be prepared to do the literature search. So I hope many of you might heard about mesh terms. Mesh is a medical subject heading, and there is a Boolean operator. So the proper use of mesh terms and Boolean operator is very much required to get the specific literature according to your objectives, right? So how to do it? If you are searching in PubMed, just click on the advanced PubMed, and in advanced PubMed, just make your query. Maybe if someone of you are working on SLR or meta-analysis, you can understand it. So you can combine that with Boolean operators and set up the proper filters. So like if you want to know the targets of rutin in the drug resistance epilepsy, so how should we set up the search strategies, right? So how to do it? So in advanced PubMed, if you are typing rutin or DRE, so what will happen? You will get lot of articles. And how you will get the articles? Lot of articles, because this search query will pick up all the articles which contain either rutin or DRE anywhere in the literature. It will include many. But what was your objective? Your objective is you want to see the literature. What is the role of rutin in drug resistance epilepsy? You don't want the article which contains only alone the rutin or alone the DRE, right? So what will be the proper Boolean operator to get this? You have to use rutin, and you have to use and instead of or. So if you are typing and instead of or, so what will happen? Only those articles will be picked up in which both rutin and DRE is mentioned. And that is your objective. What was your objective? You want to see in the literature how much literature is available with respect to rutin, the role of rutin in drug resistance epilepsy. I hope it's clear, right? So, this is just a screenshot of your PubMed. Here is the advanced options. You just click on the advanced options. So if you are using rutin or drug resistance epilepsy, so you can see the result. 19,1937 articles are there, right? Now, if you are using rutin and drug resistance epilepsy, you can see how much article has been reduced. So you get the specific articles. So again, 4,000 articles, it's difficult to read all these article full text. So next step, you have to be, read the title of the studies only. And by just reading the title of the studies, you can identify whether this article is important for me or not as per my objective. So that's why first thing is the objective of your study should be clear in your mind. Then only you can do the data mining of the literature. So you can select means by just reading the title, no need to open the abstract. You can select it. You just click on this box, it will be selected. You feel that the second article is not relevant as per my aim and objective of my studies, so you keep it unselect. So like that, you can select it, and you can create the abstract file of only those articles which you have selected. So you can select here, and yeah, in format, you can just drop down. There is the abstract text. So only abstract of those articles will be created automatically, which you have selected. Which you have not selected, that will be automatically excluded, right? So now we have AI based approaches as well. So this is a very useful tool, Rian, and it's freely available as well as the commercially available. But you can use the freely available version of Rian. So it faster the process of your literature mining. It faster the process of your data mining of the literature. So how to do it? You just create login ID and password. You can upload the articles in that one, and you can put your comment everything's on in the comment box, and you can do the blinding as well. When we are doing the data mining of literature, we have to do the blinding. At least two authors are doing it independently, so you can blind it with each other, and automatically you can see the progress as well. So this is the you can just go through this tool. There is a YouTube video as available as well. So how to use the Rian tool? It's very useful tool where you can faster the process of the data mining of the literature for your any kind of the research studies. So we have to do in network pharmacology also, we have to do the data mining, and after data mining, we have to, if we are getting any targets, we have to put in the Excel. In both Excels. Anyone has any question? So third step, after this, now you have it two files. One is the targets which can be hit by the compound, and another one is the targets which are involved in disease. So now there is a step three, identifications of the common targets. So how to identify the common targets? You can use the Vani tool. Vani tool is, again, I am just displaying the tool which are freely available. So you can just type in a Google Vani. You will find this Vani tools and you can
Excuse me,
identify
sir.
targets in the Vani. So in Vani, what you have to do, in one side you have
Hello,
to
sir. Excuse me.
targets which your drugs can be hit, your phytoconstituent can hit, whatever. You have to put in this side, and
Excuse me,
another
sir.
side you have to put the targets which are involved in that particular disease, and you will get the common targets automatically in that one. Any question?
Excuse me, sir.
Yeah.
Sir, for selecting targets, we'll receive many targets, no, sir. At that time, like what is the rationale for selecting like limited targets? Like for if we get some 100 or 150 or something,
Yes.
do we need to filter them? If we have to, then how we can do that? Based on what?
No, we are not filtering at the initial stages. At later stages, we are doing the filtrations. And filtrations we are doing depending upon the our knowledge about that particular disease. Because you know that this target have no role in this particular disease, and you are confirm about it, you can delete it. And particularly from the databases, whatever screening we are doing, we are fixing our p values. So there is a threshold values. So we are taking only those targets which are above the threshold values. So every databases have their threshold values.
Okay, sir. Thank you.
And initial stages, our purpose is to include each and every target. In later, we can do the exclusion process. But initially, don't do the exclusion process. Take all the possible targets which your molecule can hit. Explore all the databases. Whatever I have shown you, maybe you are getting one more databases from somewhere else. Include it.
Okay, sir.
Don't exclude. Include it. That's why I'm saying do the data mining of literature as well. So you have to prepare, because this is the base of network pharmacology. All the network pharmacology depend upon two Excel file. One is the target which your compound can hit, and the target which are involved in disease. Right?
Okay, sir.
Yeah.
Okay, sir.
I hope
Thank
it's
you, sir.
clear. Yeah. So now after making these two Excel file, now what is the next step? Next step is to identify the common target, common between the drug target and the disease target. That is your objective, right? You want to see how my molecule can act as an anti-cancer molecules, or how my molecule could play a significant role in the diabetes. What is the possible mechanism? So to get this, now you have a two Excel file. Now you have to identify the common targets. So in one side, you just paste the drug target from where you got this drug target, whatever the Excel you have prepared. And in this one disease target, whatever the Excel you have prepared for the disease target, DigiNet, OMIM, all these databases, right? And now you can see the results. So in this case, we have found the 35 common targets, which are the target which are common in the drug and in the disease. Now next step is step 4, interaction among the targets. So now you got the common targets. Now you can see how much it's refined, whatever the question you
Hello,
have
sir.
asked, how you will refine it. So now you can see only 35 targets. You are going for the next step. Got it?
Hello.
Yes. Hello.
Sir, as you say, we can get common targets. Sir, I was working on that some days ago, and from common targets between disease and my phytoconstituents,
Yes.
I got one particular target. But when I do literature search regarding that, it says that it is not directly involved in the progression of the disease. What I can do in that case?
You have to include it.
What, sir?
You have to include it.
But sir, it is saying not involved in the progression of the disease.
Yeah, it's
It is
not
the
involved in the progression of disease that
yes, sir.
you in the last step you can exclude it, but at initial step include it.
Okay.
Hello,
Yes, yes, sir. Thank you, sir.
got it?
Yes, sir. Yes, sir. Thank you, sir.
So initial steps you include each and everything. There is a we are applying the VLOOK formula as well in the Excel. So in that one we are highlighting only those targets which have a role in that particular disease. So in that step we can exclude others, but in these steps you have to include each and everything. Okay. So next step
Oh, oh,
is interactions among common targets. So next step is, and the string is widely used, you just type in a Google string, you will find it. And string is, established by European laboratory in 2019, and it provides the combined score is computed by combining the probability from the different evidence channel and correcting for the probability of randomly. So cutoff value we are keeping 0.7. So setting the cutoff lower, if you are keeping it lower, so will increase the coverage. It will increase your coverage, but also increase the chances of false positive errors, right? So you have to be very careful when you are changing the cutoff values. So this is the string database. You just type in a Google string. This is the screenshot of string database. And in string database, you just select the multiple proteins and just add it. You can get the interactions among the targets. So you just here also you can select the organisms, and then you can get the interactions among targets, how targets are interacting with each others. So that you can do it. And again, I am getting your meeting will end in 10 minutes. Okay. So this is the Excel work which we have to do. I have added the screenshot. We have to be arranged, and whatever these yellow highlighted I have done. These are the targets which I confirm. This is the targets which are involved in the disease, right? As someone of you have asked, how we will assure that we are, means if we are getting the targets which are not involved in that disease. So you have to do these screening, filtering process here in the Excel. So you can select only those which you are confirmed, right? So these are the small steps we have to do it. We have to arrange these data. And then for the interactions, there is a BioGrid is also available, and it's also freely available. And this is the web page. So welcome protein, genetic and chemical interactions we can do by BioGrid as well. There is a DIP is another one. DIP also we are using to check the interactions among the targets. So this is the web page of the DIP. And there is a Intact. Intact is another one which is supported by the European Molecular Biology Laboratory core funding. And this is the web page of the Intact Molecular Interactions database. So, of course, we have to do, as I said, little bit sorting. So earlier we are also using the VLOOK formulas in the Excels to identify the common targets and important targets, which we can by using them, we can create a network by using the cytoscape. So step 5 is the creation of network, and creation of network. Cytoscape is the most commonly used. You can download it. It's freely available. Just type in a Google cytoscape and you can get the cytoscape, and in cytoscape you can upload the files and you can create the network. So you can see there is a visualization process as well. There is a analyze network option is also there. You can analyze various parameters, node, betweenness, how, whatever I have shown you in earlier slides. So you can do the analysis and you can display the network in circles or other way as well. There is a cytohuva as well where you can identify the most important targets as well. So there is a CAG. CAG is, how to fully displayed information combined in a cells with the help of computer. So CAG is expand for Kyoto Encyclopedia of Gene and Genome, established by Cancer Laboratory of Bioinformatics Center of Kyoto University in 1995. and need to predict complex cellular pathway based on information in the genome. So as I said, from the KEGG, you can identify the possible pathways. So this is a KEGG enrichment analysis. You can do it by gene enrichment analysis, bubble plot, enrichment bubble. If you are doing it, so you will get the pathways. Because earlier steps from the cytoscape, you are just getting the targets. So pathways, because these targets will make some pathways. So pathways, you can do the KEGG enrichment analysis. There is a Sinogeo as well. From there, you are also can do the these signal transductions. You can get it about the pathways informations. Then, as I said, after getting these all doing all these steps, these will not tell you about how your molecule is interacting in the active site of the target. So to check that, you have to do the molecular docking. And docking will predict the energetically favored binding conformation of the ligand in the active site of the receptor. So I hope nowadays docking is very popular and very common. So many of you knows the process. I'm not going in detail, but there are a standardized steps where you have to first prepare the structure, select the target, preparation of target, then grid generations, then you can go with the HTS, SP or XP. Yes, anyone is saying something? So to get the structure of the targets, you can search in the PDB, and in the protein data bank also there is a criteria for the selection of the targets. Because if you are adding the name of any targets, suppose acetylcholinesterase, you will get number of proteins are available in the protein database. So now question is which one you have to select. What are the what are the criteria for the selection of the PDB? So there is a resolution value. There is a you have to look for the mutations. You have to look for the internal ligand is there or not, right? So you have to prepare the ligand. This is just a screenshot which we are in our lab. We are using the Schrodinger. So we are preparing these target preparations, and then we are generating the grade. If internal ligand is not there, we are doing the site map. Then we are performing the docking studies, and then we are doing the MMGBSA calculations, binding energy, ligand complexes by using molecular mechanics, generalized bond surface areas. We are calculating it. Then next steps, we are doing the stability of the drug or new chemical entity in the active site of the targets. And that I said molecular dynamics, which depends upon the Newton's second law. That is the force is equal to mass into acceleration. And acceleration is velocity, change in velocity with respect to time. And these are the steps which we are doing in the GROMAC. GROMAC is a freely available, but of course, little bit command-based. Linux is required, but it's freely available. By that one, you can do a lot of parameters as well. But to do the molecular dynamics, your system should have a graphical processing systems you should have. In normal CPU, it will not work, right? Because many research studies are asking you about 100 nanosecond simulation, some are asking about 200 nanosecond simulation. So you should have a high performance computer systems. Then system build up molecular dynamics, which we are doing, and these are the parameters which are we are calculating. RMSD, root mean square, RMSF, root mean square fluctuations of amino acids, SASA, number of hydrogen bonds, radius of gyrations, then free energies, then PCA, principal component analysis, then free energy landscape as well. So after this, you have to be plan. This is all up to that, all the computational part. So next part, you have to be design the experimental studies. So experimental study, depending upon your resources, you can plan for the in vitro studies. So you can plan for the cell lines or cell culture studies. So whatever the target you got through computational systems, you can validate it experimentally. You can expose the cells with your drug concentrations at different concentrations of the drug, and then measure that particular proteins. And to do the proteins, I think every one of you knows you can perform the Western blot analysis. So you can check the expressions of the proteins after the treatment, before the treatment, and you can compare your, you can connect your computational results with the experimental results. If you don't have a in vitro cell culture facility, so you can directly do the animal experimentation and study as well. And after giving the doses to the animals, you can measure. You have to do the so again Western blot or RT-PCR. If you are planning to do the gene analysis, you can do with the RT-PCR. Right. So recent work done, what work we have done in our lab at DIPSA, DIPSRU with respect to that. So this is the one work which we have done, repurposing of the drugs against hepatic encephalopathy using pharmacovigilance based approaches integrated with experimental verification. In that one, what we have done, we have applied data mining algorithm first on post-marketing data, and data we have collected from the FAIRS database. FAIRS is the one of the most commonly used database. That's the FDA Adverse Event Reporting Systems. And we have analyzed that data. We have calculated PRR, ROR, and Bayesian approaches, and by that one we have identified the novel this one inverse signal approach, and we have identified the molecule which can be repurposed for the pharmacological results. These are the network pharmacological approaches where we have identified what are the possible targets which can be hit by the molecules, and then we have performed the molecular docking and dynamics. And then finally, we have done the animal experimentation studies. And then we have done these histopathological or these, SGOT, SGPT in livers and ammonia. These all we have measures. And we find that atenolol and amlodipine can be repurposed for the hepatic encephalopathy. Hepatic encephalopathy is something, it's a serious conditions, and current treatment options are very much limited. We don't have a specific drug for its treatment. That's why we have planned to repurpose the molecule for the hepatic encephalopathy. So in this case, what we have used, first we have used the pharmacovigilance data. On pharmacovigilance data, we have used data mining algorithm. Then we coupled with the network pharmacological approaches. Then we done docking studies, then dynamic, and then experimental animal studies. And this is another work. This is the same work. So these pathways we have, identified in this particular case. So you can see MAP kinase and PI3-AKT pathways. And the molecule which we have identified is amlodipine and carvedilol. Carvedilol can inhibit your MMP9 and NF-kappa B. Amlodipine can PARP1 as well as NF-kappa B. So if these are going to inhibit, your inflammatory cytokines will decrease. Then another work which we have done on the drug resistance epilepsies, we have screened a lot of natural molecules which are available on natural product databases, where we have screened against various targets of drug resistance epilepsy. Then you can see there is a 42 targets which compounds can hit. There